I, Dr. Vanangamudi, welcomes all the students and teachers for the presentation on self-incompatibility, that is part 2, on behalf of Trinity Cultural Academy, Coimbatore, and New India Publishing Agency, New Delhi. And in the last video, we have seen about the part 1 of self-incompatibility, and here the part 2 self-incompatibility will be explained to you. And as I mentioned in the earlier presentation, there are 5 types of self-incompatibility. In the previous series, we have seen the single locus self-incompatibility, that is gametophytic self-incompatibility and sporophytic self-incompatibility. And in this presentation, and we will be explaining to you about two locus gametophytic self-incompatibility, heteromorphic self-incompatibility, and cryptic self-incompatibility, and late acting self-incompatibility. We have already seen these two types, that is gametophytic self-incompatibility and sporophytic self-incompatibility. And with respect to two locus gametophytic self-incompatibility, it is mostly available, exist in the grass subfamily Phoididae, which have gametophytic self-incompatibility that involves two unlinked loci referred to as capital S and capital Z. If these alleles expressed at these two loci of pollen grain both match the corresponding alleles in the pistil, and the pollen grain will be recognized as incompatible. And that's all about two locus gametophytic self-incompatibility. And the third one is heteromorphic self-incompatibility. Here the genes responsible for self-incompatibility in heterostylous flowers are strongly linked to the genes responsible for flower polymorphism. So these traits are inherited together. And there are two concepts. One is distyly and tristyly. Regarding distyly, here both stamens and styles are of two types. The stamen may be low or high, and styles are short and long. It is determined by a single gene with two alleles. And there are two types. That one is thrum type and pin type in distyly. The thrum means the flower with the short style, and high stamen is called thrum type. And the with regarding to pin type, the flower with long style and short stamen are called the pin type. Both thrum and pin flowers differ with 6 characteristics in addition to stamen and style length. And the incompatibility mechanism in distylys are explained in this table, and it is the same genotype, the thrum and having both the capital S and small s and capital S and small s. If both the similar genotypes are crossed, there is a incompatible. The pollen grain will not germinate on the thrum type. Similarly, the smallest of pin and the same genotype, the smallest of pin, and the pollen of this parent will fall on the stigma of the same genotype. There is incompatibility. And if both genotypes are pollinated, like the capital S, small s thrum pollen is fall on the smallest pin, or smallest pin pollen is pollinated on capital S gametid small s thrum, that is heterozygous, this is homozygous. Both the genotypes are different. 1:1 compatible mechanism exist, and the SS smallest pin, this is a female parent, and capital S and smallest thrum, if the pollen of this type, this plant falls on the different different flower of the different genotype of the stigma, there is a 1:1 compatible mechanism exist. And regarding Tristyly, the styles and stamens have three different positions, and these two genes are involved, the capital S and capital M, with two alleles, and capital S gives rise to short style, capital S and capital M to medium style, and small s and small m to long style. This is a long style flower, and this one is the medium style flower, and the third figure is the short style flower. Third flower is the short style flower. And as far as fourth type is concerned, this is a cryptic self-incompatibility. It is existing in a limited number of crops. The best examples are bladder campion, Silene vulgaris, the family is Cariphyllaceae, Viper bugloss or blueweed, Echium vulgar, the family Poroginaceae, and the water willow or swamp loose strife, that is the Decodon verticillatus, the family Lythraceae. This is the bladder campion, and this is the water willow, and this is vipers, bugloss or blue weed. In this mechanism, the simultaneous presence of cross and self pollen, the simultaneous presence of cross and self pollen on the same stigma results in higher seed set from cross pollen than self pollen. However, the cryptic self-incompatibility mechanism, self-pollination without the presence of competing cross-pollen, that results in successive fertilization as well as successive seed set. In some species, pollen tube elongation leads to faster elongation of cross-pollen tubes than self-pollen tubes. Ratio of crossed to selfed ovules ranges from 3:2 and 11:5 when equal amounts of cross and self pollen are placed upon the stigma. And the fifth one, last self-incompatibility type classification, is late acting self-incompatibility. It is also termed as ovarian self-incompatibility. In this mechanism, self-pollen germinate and reaches the ovules, but there is no fruit set. Absence of fruit set is due to genetic factors, that is homozygosity of lethal recessive alleles, which are the direct result of self-fertilization, that is inbreeding depression. LSI can be pre-zygotic, that is a deterioration embryo sac prior to the pollen tube entry in Narcissus trianthus. This is the Narcissus trianthus flower, the plant. And the another type is post-zygotic, malformation of the zygote or embryo. Example, Asclepias and Spathodia campanulata. This flower is Spathodia campanulata. It is mostly available, commonly available as avenue plantation. And the last aspect is the maintenance of self-incompatibility, how it will be maintained in breeding program. And there are four types. The first one is bud pollination. It is supposed that some compound which inhibits the pollen germination and tube growth is produced by stigma and style at the time of flowering. There is a inhibitory substance in the stigma and style that will inhibit the pollen germination and tube growth. If pollination is done 24 hours before the natural opening, there is incompatibility. At this stage, probably there is no inhibitor compound is produced, and widely it is used in grapes, cabbage and cauliflower. In these crops, the lines are maintained. Self-incompatibility lines are maintained by bud pollination. The third one is end of season pollination. Usually when the crop season is about to end, at the end of the season, incompatibility mechanism weakens, and this probably due to less metabolic activity of the plant, and there must be a failure of the plant to produce enough inhibitory compounds that will inhibit the pollen germination and tube growth. And the third one, the stigma removal. In cauliflower, cabbage and radish, failure of pollen to germinate to tube growth occurs. So removal of stigma and then pollination results in perfect seed set. And the fourth one is grafting in trefoil parency. Grafting of vegetative pods either within a single plant, that is homograft, or between the different plants, that is heterograft, have also improved the level of compatibility. Pollination can be done by grafting, making polyploidy of incompatible strains. They may become compatible because of altered genic balance at the tetraploid level. This method is being fully utilized in tree fruit crops like peach as well as in potato. And regarding the importance of incompatibility in plant breeding, and the self-incompatibility fruit trees, when they are planted at the ratio of two cross-compatible varieties to ensure fruitfulness. And self-incompatibility may be used in hybrid seed production very extensively. For that hybrid seed production, two self-incompatible but cross-compatible lines are to be interpolated, so the seeds obtained from both lines would be hybrid seed. And self-incompatibility provides a way for hybrid seed production without emasculation and without resorting to the mechanisms like the genetic and cytoplasmic male sterility mechanisms. The self-incompatibility system permits combining of desirable genes with a single genotype from two or more different sources through natural cross-pollination, which is not possible in self-compatible species. In pineapple, since commercial clones are self-incompatible, the fruits produced from these lines are parthenocarpic, that is seedless. And there are some limitations. It is very difficult to produce homozygous inbred lines in a self-incompatible species, but pollination has to be made to maintain the parental lines. Incompatibility is reduced or broken down at high temperature and humidity, so the environmental condition also plays an important role in deciding the self-incompatibility mechanism. There is a limited use of self-incompatibility, very limited use due to problems associated with the maintenance of inbred lines, that is the homozygous lines, through hand pollination. Yes, it is very tedious process and also very expensive. Thank you very much for listening this presentation on self-incompatibility part 2, and kindly watch this video, and you watch this and also give your comment, valuable comment, so that we can improve our presentation. Kindly subscribe to this video for getting future videos at regular intervals.